The COVID-19 pandemic drew attention to the importance of accurate, accessible, and rapid testing in response to emerging infectious diseases. In this video, we'll be exploring SHINE, an innovative diagnostic tool designed to address these needs. While the assay can be adapted to different pathogens, we are presenting it here as part of a public health initiative to detect Lassa Fever in West Africa. RT-qPCR, which is the gold standard diagnostic for Lassa fever, requires specialized laboratory equipment and staff. While RT-qPCR gives accurate results, it takes 2 to 4 hours from sample preparation to the completion of the qPCR run to obtain a result. SHINE, which stands for Streamlined Highlighting of Infections to Navigate Epidemics, employs CRISPR-based diagnostics and lateral flow test strips to provide an equipment-free and a faster result with a colorimetric readout. Notably, instead of cycling through multiple stages of heating as with traditional PCR, SHINE uses a constant temperature to make copies of the target sample material by what is known as isothermal amplification. For SHINE, this is at 37 degrees Celsius, our body temperature. All reagents can be lyophilized or freeze-dried in advance to retain stability if it needs to be transported long distances for clinical testing. After watching this video, you will have a foundational understanding of the step-by-step process of SHINE. Specifically, you will 1. Learn about how SHINE works for the detection of a specific pathogen. 2. Understand the molecular mechanisms behind a CRISPR-Cas lateral flow assay, or LFA. And 3. Identify the step-by-step process from sample collection to test strip readout and interpretation. To diagnose Lassa fever, patient samples are collected through a blood draw. Since the Lassa virus is a single-stranded RNA virus, we start by extracting all of the RNA in the sample. Next, lyophilized SHINE reagent pellets are resuspended in solution using a rehydration buffer. Of the many components in the reagent mix, key elements include 1. Primers designed to bind to the Lassa virus sequence, 2. T7 RNA polymerase to make RNA from cDNA, 3. Lassa-specific Cas13 CRISPR RNA complexes that cleave single-stranded RNAs when activated, and 4. FAM biotin reporters for the lateral flow colorimetric readout. Once the sample is added to the reagent mix, the resulting solution is incubated for 90 minutes at 37 degrees Celsius for the amplification and the CRISPR-Cas13 cleavage steps. If the Lassa virus is present in the sample, the primers will bind to the virus-specific sequences and generate millions of cDNA copies through a process known as reverse transcription recombinase polymerase amplification, or RT-RPA. No such copies will be made if the Lassa virus sequence cannot be found in the sample. Next, T7 RNA polymerase transcribes the cDNA copies back into RNA. Lassa-specific CRISPR RNA guides the Cas13 protein to the specific RNA sequence of the Lassa virus, forming a complex. The binding of this complex activates the cleavage activity of Cas13, cutting both the viral RNA and nearby RNA reporter molecules. If no Lassa virus is present, the reporters remain intact. The higher the concentration of Lassa virus in the sample, the more reporters are cleaved. After the LFA buffer is added to dilute the reaction, finishing up the assay is simple. Insert the test strip and results can be read in about 5 minutes. But how does this work on a molecular level? Let's rewind and take a closer look at the inner workings of a lateral flow assay. First, let's see an example where the Lassa virus is present. So, many of the reporters have been cleaved by Cas13. Each reporter consists of a FAM end, which specifically binds to anti-FAM antibodies, and a biotin end, which binds to a protein called streptavidin localized on the control band. As the sample flows up the strip, it resuspends the gold nanoparticles from the conjugate pad. These particles hold the key to the color change for visual readout. They contain anti-FAM antibodies that bind to the FAM ends of reporters. When the sample reaches the control line, the biotin ends of both cleaved and uncleaved reporters can bind to the streptavidin on the band. However, only the uncleaved reporter, which consists of both the biotin and FAM ends, has a bound gold nanoparticle, which causes the line to turn red. When the FAM ends of the cleaved reporters reach the test line, the anti-FAM antibodies of the gold nanoparticle binds to the anti-conjugate antibodies, which turns this band red. If the Lassa virus is not present, all of the reporters remain intact and bind to the control line, which turns red. This colored band confirms that the solution flowed correctly across the strip, indicating the test's validity. However, in the case where there is a very high concentration of the Lassa virus present, there may be no intact reporters left to attach to the control band. So, a missing control line doesn't always mean the test is invalid if the test line is red. After the sample flows up the strip, it is removed from the tube and photographed to capture the results. Here, you can see the three main readouts: negative, positive, and inconclusive or invalid, alongside their corresponding band patterns. So let's recap. SHINE involves 7 main steps. 1. Collection of patient sample. 2. Extraction of target from sample. 3. Rehydration of reagent pellet. 4. Addition of target to reagents. 5. Incubation at 37 degrees Celsius for RT-RPA and CRISPR-Cas13 reaction. 6. Insertion of test strip into the reaction mix. And 7. Interpretation of results after 5 minutes. In summary, SHINE is a lateral flow detection platform where the sample and reagents are incubated together. Utilizing CRISPR-Cas13 technology, the results are displayed colorimetrically after inserting the test strip into the reaction mix. SHINE can be easily modified to detect diseases other than Lassa fever by incorporating different pathogen-specific primers and CRISPR RNAs. Ultimately, SHINE can be used for point-of-care pathogen detection to facilitate widespread testing in the event of an outbreak. We hope that you enjoyed this video and found it helpful to learn about how SHINE can be used for rapid diagnostic testing. To learn more about outbreak response, check out the rest of the videos in our series. Special thanks to our dedicated team of researchers and the Broad Institute video production team who supported the creation of this video.